Howdy, folks. Thanks for checking into Mr. Ulrich's LandofBiology.com. I am Mr. Ulrich. Now, those of you who have me in class know that I really enjoy exposing students to new musicians that they may have never heard of before. And as much as I'd love to listen to Yellow Man or the Edgar Winter Group, that's not really what this Nosecast is about. However, those musicians and the Arctic Fox all show patterns of inheritance that don't really follow the rules, so they're all lawbreakers. So this Notescast, we're talking about breaking the laws. Remember from our previous video in Mendelian genetics that he was kind of lucky in that he chose traits in the peas that were pretty simple patterns of inheritance. They were all monogenetic in that they were controlled by one set of genes. There were only two alleles in the population, and one was always completely dominant over the other one. But now we know that it's just not that simple. One of these patterns of inheritance that doesn't follow basic Mendelian laws of dominance is incomplete dominance. In this case, the heterozygote shows an intermediate phenotype, a third different phenotype that's kind of a little bit of either of the other phenotypes. The example here in carnations is the red carnation is homozygous dominant for red, makes a protein that makes the red pigment. The white flowers don't make that pigment, so they don't have a gene that codes for that pigment. The heterozygote makes a pink flower. The pink is actually a result because it only makes half as much of the red pigment. So it does show a third phenotype in between. The way we write this is not using capital and lowercase letters. We would say that the red flowers were homozygous dominant red, the white flowers were homozygous dominant white, and the pink flowers were a mixture of both heterozygous red and white. We take a look at the classic homozygous red versus homozygous white test cross. We get the F1 generation are all hybrids. The homozygous red can only donate red alleles to their offspring, and the homozygous white can only donate white alleles to their offspring. So not very interesting there. Where it gets interesting is in the F2 generation. If we do the self-pollination, we end up with a different, not a 3-to-1 ratio, but a 1-to-1 ratio. Let's check out this ratio by doing a quick Punnett square of that F2 generation. So of course we have two parents, both of which are heterozygous. So either parent can either make gametes that carry the red or the white allele. And when we go through and do the good old Punnett square, we see that we have 1/4 of the offspring will be red, 1/4 will be white, and 2/4 will be pink, the in between. So hence the 1-to-1 ratio. Another pattern of inheritance that doesn't follow basic Mendelian laws of dominance is codominance. In this case, we can still have two alleles, but they both code for a working protein. Remember, with incomplete dominance, we still have two alleles, but one codes for a protein, the other one does not. The difference is that the expression of the protein, the expression of the phenotype, has to do with the amount of genes or the number of alleles that you have. So if you are a homozygous individual, homozygous red, you have two alleles as opposed to a heterozygous who only has one allele. So the homozygous red makes twice as much pigment as the heterozygote does. In codominance, the heterozygote is not a blending. It's not an intermediate between the two. You get a full expression of both alleles. How does this work? Well, one example is roan color in horses and cattle. In this case, this is a blue roan horse. It is the heterozygote between a black-haired individual and a white-haired individual. And when we look at each individual hair, each one is either black or white. You don't have gray hairs, the intermediate between them. The result of this kind of salt and pepper juxtaposition of the black and white hairs is a kind of this bluish color, which is kind of neat. In humans, we see this in the ABO blood typing system. There are 3 alleles present in the population for blood type. Each person still has 2. We're still diploid. It's just the possibilities are a little bit more with the 3 alleles. There's the A allele, which is codominant with the B allele, which are both dominant over the recessive O allele. Gets the letter I. It's lowercase there. In this case, like I said, the A and B alleles are co-dominant. That means that they code for a glycoprotein in this case. And individuals who are heterozygous for A and B make both A glycoproteins and B type glycoproteins. Remember those glycoproteins, they make up the carbohydrate trees. That's that forest on the outside of each cell. The A and B antigens on red blood cells make up just one or two of the different types of trees that can be out there in that forest. The O allele is recessive to both because it codes for neither an A nor a B antigen. That doesn't mean that individuals that are O type don't have any antigens. It just means that they don't have either the A or the B antigen. The genetics of the ABO blood typing system in human not only gives us a good opportunity to talk about codominance as well as multiple alleles, but we can throw around these terms genotype and phenotype, and we can even talk a little bit about the immune response. So individuals who are type A blood, so they're phenotype A, only display the type A antigens on the surfaces of their red blood cells. Now, I'm only talking about type A versus type B antigens. All right, there's still that whole forest of other antigens, other glycoproteins, signaling glycoproteins on the outsides of the cells. We're just talking about focusing on these two. All right, so in order to just have the A antigens, you can only have the instructions for those A antigens, so you have to have just the A alleles. And there are two genotypes that are going to yield that particular A phenotype. You could be homozygous A or heterozygous A, where the second allele is the recessive O allele, because that doesn't code for either A or B. So those individuals with those genotypes both have just the instructions for those type A antigens. Now, where the immune system comes into play here is you are born with antibodies against the type A or type B antigens that you don't have. That means that a type A individual is going to have anti-B antibodies. That means that if any blood gets into their bloodstream that displays the B type antigen, the anti-B antibody will stick to that blood cell and stick to other antibodies and clumpy, and they all stick together, and that's no good for circulation. So we got to be careful when we mix blood. Now, the phenotypically B type individuals, again, can have the same two types of genotypes, homozygous B or heterozygous B, with that recessive allele being the second one. They're only going to display the B type glycoproteins on the outside of their red blood cells and hence have the anti-A antibodies. Now, the heterozygote AB individuals, this is where things start to get interesting. Since A and B are codominant, just like the hairs in horses, in roan horses, are either fully black or fully white, the antigens on the surface of type AB individuals are either type A or type B. That means they can't have either anti-A or anti-B antibodies, or they'd be autoimmune, they'd be reacting to themselves. So though this says no antibodies, don't be thrown off. You still make antibodies, you AB people, just neither anti-A nor anti-B. This means that if anybody's blood gets into a person who is type AB, because they lack the antibodies, anti-A or anti-B, there's going to be no negative response, and that's going to be a safe transfusion. So people who are type AB are the universal recipients. Problem, of course, is they have both the A and the B antigens, so they can't be donated to really anybody because their blood will react with A's anti-B's, B's anti-A's, and type O's, both anti-A and anti-B, as we're going to see here in a second. So jumped ahead, type O is homozygous recessive, so they display neither the type A or type B antigens. This says no antigens. They still have all the other antigens, just not the type A or type B antigens. What they have, of course, are both anti-A, like I said, and anti-B antibodies. So they can't take blood from anybody other than type O people. However, they can donate to anyone because they lack the antigens on the outside of them that will respond with other people's anti-A or anti-B antibodies. So they are the universal donor. So far, we've been talking about genes coding for proteins or not coding for proteins in the case of recessive alleles. And those proteins or lack of the proteins having a particular effect on the phenotype. This is a bit of an overgeneralization. What we know is that most of these proteins can have a range of effects depending on where in the cell they are, how much of them there are, when they're produced during development, a whole host of variables. So that means that most of these genes, therefore, are we call pleiotropic, and that they have lots of different effects. That means also that if there's a problem with one of these pleiotropic genes, there's a wide range of phenotypic implications of that mutation. Dwarfism, the achondroplasia version of dwarfism anyway, is a single mutation in one gene. It codes for a growth factor receptor molecule that normally shuts down or negatively impacts long bone growth. And so these individuals have shorter long bones, so they have shorter arms and legs. Marfan syndrome is also a single mutation that codes for a protein that's involved in connective tissue. And there's a wide range also of phenotypic effects from circulatory issues to eye problems to ligaments and tendon problems as well. Let's take a look at the pattern of inheritance seen in achondroplasia. It does follow dominance in that individuals who have achondroplasia, like Peter Dinklage, have one dominant allele. His wife, Erica Schmidt, she is of typical height, typical stature, so she's homozygous recessive. If we compare their Punnett square and their expected ratio, phenotypic ratio of offspring to a couple who both have achondroplasia, we see a little skew, we start to move away from typical Mendelian genetics. Looking at Mr. Dinklage and Ms. Schmidt, at their Punnett square, she's going to make or has made eggs with either the recessive or the recessive allele. Not that exciting. Half of Mr. Dinklage's sperm, they'll carry the dominant achondroplasia allele. That means, when we do the Punnett square here, that they have a 50% chance with each child of having a baby with achondroplasia. Now, in the couple where both of them have achondroplasia, where their Punnett square is going to look like a typical monohybrid cross. And though we would expect to see a 3-to-1 ratio of achondroplasia to typical, we don't. And that's because this homozygous dominant combination is lethal. Without at least one recessive allele, the zygote is non-viable. And so those never come to term. And so when we see the pattern of offspring, we see the phenotypic ratio of offspring, we actually see a 2-to-1 ratio instead of a 3-to-1 ratio. Epistasis is another mechanism or pattern of inheritance where we're going to end up with a phenotypic ratio of offspring that's a little different from what we would expect through basic Mendelian patterns. Coat color in mice is a good example of this, where there are 2 separate genes that we're talking about here. So again, we're talking about almost a dihybrid cross, so get those hats on. In this case, one of the pair of alleles codes for pigment or no pigment, and then the other pair of alleles modifies the amount of pigment that's made if it's made. So if you have no pigment, then it doesn't really matter what the other alleles are as far as modification goes, because if you're not making any pigment, then it doesn't matter if you're going to make more or less of it. So we end up with 3 phenotypes. If you have a dominant allele at both of the loci, then the black coat color is seen. If you're recessive at the more or less pigment locus, but you have pigment, that's where you get the brown coat color. But again, it doesn't matter what that modification allele is, as long as you are homozygous recessive for pigment, you're going to be an albino no matter what. So here's where we see, instead of the 9:3:3:1 ratio, we get a 9:3:4 because the last 3 and 1 are combined into one group of albinos. We see the same pattern of epistasis in Labrador retrievers. It's actually set up the same way, with 2 sets of alleles, one set coding for pigment or no pigment, and the other set of alleles modifying the amount of pigment that's made if it is made. So we see the same kind of setup in that we're going to have yellow labs instead of albinos here. But then we'll also have chocolate labs for the low expression of the pigment and black labs for the high expression of the pigment. We've been talking about traits that are controlled by one set of alleles or maybe 2 sets of alleles. Most traits are actually the result of lots of different proteins acting together in concert. So they're actually influenced by many, many different types of, or many different sets of alleles. We call these polygenic traits. And the phenotypes will exist on a continuum, on a kind of a sliding scale. Instead of having red or pink or white, we have lots of all of the grades in between. When we look at things like skin color or height or weight or intelligence or even behaviors, all of these occur on some sort of continuum, which means that each one of us, the only individuals that have ever had that particular grouping of genes, unless you have an identical twin, making us all snowflakes. Sure, skin color exists on a continuum, is influenced by a whole host of genes, upwards of 7. However, just like in epistasis, a single pair of those alleles can mask all of the other genes that are involved. In order to have skin color, you have to be able to make melanin, or hair color, or eye color for that matter. All of these rely on this one pigment. In order to make that pigment, you need an enzyme to convert the tyrosine into melanin. Individuals who are homozygous recessive for this enzyme don't make the enzyme and therefore don't make melanin and are true albinos. It is pleiotropic in that melanin is needed for lots of different phenotypes. So it's not just skin color, it's also hair color and eye color, and even some issues with vision are involved. Well, we'll stop there. I think that's enough for now. We'll move on to sex linkage and environmental influence on gene expression in the next video, and that'll continue on the same notes sheet. Again, thanks for checking into Mr. Ulrich's Biology.com, and if you have any questions, please feel free to send me an email or leave a message in the comment section below. Other than that, we'll see you in class.